Thank you for the invitation. My name is Min Cho. I'm an assistant professor at Johns Hopkins. Today, I'll be talking about neuropathology in COVID-19. These are my disclosures. Objectives. So we'll be talking about neurologic symptoms and complications, and some virology, and how SARS-CoV-2 enters the brain CNS system. And we'll be talking about quite a bit of time on data on detection of SARS-CoV-2 virus in brain tissue. And we'll discuss different pathological findings in this talk. As we know, there are a lot of patients with COVID-19. I think a lot of different talks are discussing different neurologic complications, how burdensome this disease is. So we'll skip this part. If you look at the schematics, many neurological symptoms were described over last 1.5 years. I would like to emphasize that the causality has not been established in these neurologic complications and COVID-19. You can see in the diagram, a lot of different diseases have been described. And let's have you focus on this picture schematic. I highlighted 2 question marks here. So when SARS-CoV-2 enters the CNS system, there's 2 theories. First is olfactory neuroepithelium, how virus can go through the nasal cavity and go into the frontal lobe, and if that's the cause of a CNS infection. And the second most common theory is to have bloodstream infection spread, and the viremia and getting into the bloodstream and going into the CNS system. So we'll be talking about this different entry hypothesis, and the fact that anosmia is pretty common is interesting. That that olfactory pathway may be actually the cause of CNS infection, but we'll be talking about this in neuropathological studies. So SARS-CoV-2 coronaviruses are single-stranded positive sensed RNA virus, and it's a beta coronavirus, and it shares genetic similarities to SARS-CoV-1 and also middle MERS-CoV virus, 79% homology and 50% homology in each virus. And it's got 4 proteins. As you guys probably know about this, there's envelope membrane, nucleocapsid, and spike glycoprotein. And these 2 proteins are pretty important in immunostaining to get this virus tested. Let's talk about transcribial entry to the CNS. This is a most popular theory how SARS-CoV-2 enters the CNS system. So neuronal entry of SARS-CoV-2 relies upon spike protein, as we know, and as you guys probably know, that ACE2 inhibitor, ACE2 receptors are really key receptor to go into the CNS system. And the cleavage of viral spike protein by this receptor, transmembrane serine protease 2, TMPRSS2, is known to activate the virus. And ACE2 mediated CNS entry into olfactory sense neurons, OSNs, of the olfactory epithelium, and then that enters second order neurons along the olfactory nerve, and finally brain tissue. So that's the very popular theory, and that's why we think anosmia is very common in these patients, and maybe a fraction of patients may have CNS entry after that. And this schematics shows that how close it is between the cribriform plate from the olfactory sensory neuron or nasal cavity to basal frontal lobe with olfactory bulb. And you can see the nerve here that connects basal frontal lobe from the nasal cavity. So there is a plausible mechanism here that transcribial spread may be the cause of CNS entry for SARS-CoV-2 virus. I just wanted to show you that location of ACE2 receptors in the brain, usually medial system, and you can see in the frontal lobes, motor cortex, and some cingulate cortex, and ventricles. Choroid plexus is a popular has abundant ACE2 receptors, and the lower brainstem is another popular location for neuropathological findings for SARS-CoV-2. And I think these locations are pretty important when we talk about CNS entry to the SARS-CoV-2 virus. Let's talk about other data. So if you look at some MRI data, you can see in the first schematic MRI of patients with olfactory dysfunction and anosmia and COVID-19 infection. You can see corona brain MRI with the T2 imaging here, showing the thickening and edema of olfactory mucosa and visual atrophy of olfactory bulb in this patient. So there's MRI evidence of olfactory changes after COVID-19 infection. And the second schematic shows really basal frontal lobe olfactory bulb of the CNS system, showing hyperintensity in the MRI. This is a JAMA Neurology paper showing that COVID-19 infection can cause hyperintensity or damage to basal frontal lobe with the infection. When this patient also had anosmia. And the last picture is basically showing that the SARS-CoV spike protein antibody, when you stain it, you can see virus in this staining in the pathology for these patients with epithelial cells and in the nasal cavity. And you can see if you do in situ hybridization, SARS-CoV-2 RNA can be picked up in the mucosa layer and cells of the epithelium. So really showing that nasal cavity really shows the pickup of SARS-CoV-2 when you stain this immunochemistry or ISH technology as well as MRI findings. Let's talk about bloodstream infection or viremia spread. So another popular theory is viral invasion through the BBB and also blood CSF barrier through the endothelial cells. Frequency of viremia in COVID-19 patients varies widely, 1 to 41%, pretty significant wide ranges. And theory is circulating SARS-CoV-2 can bind to BBB or brain or blood CSF barrier endothelial cells, where host proteases cleave SARS-CoV-2 spike protein into S1 and S2 segments. And this spike protein S1 fragment crosses the BBB by ACE2 mediated transcytosis. And also SARS-CoV-2 spike protein can trigger immune activation, and it increases BBB breakdown and permeability, which also exacerbates or really helps to get into CNS system from the COVID-19 or SARS-CoV-2 to the CNS entry. And SARS-CoV-2 can also infect mature ACE2 expressed human choroid plexus cells in vitro. And although there was no viral spread to proximal cortical regions, despite 44% reduction in blood CSF barrier integrity. And another theory that's floating around is infection of vessel wall, and which is a kind of encephalitic process, but this has not been studied well and not documented well, but there is some theory about that as well. So early studies, if you look at this Nature Reviews in Neurology early in 2020, a lot of different rates or frequency for neurologic symptoms of COVID-19. Histopathology, this particular review had at the time reviewed a couple papers reporting histopathology, which is pretty non-specific, acute hypoxic ischemic injury, hemorrhagic, and lymphocytic paraencephalitis, meningitis. But after this study, there were a lot of studies coming out. We'll go over a couple studies that important studies highly cited for neuropathological features of COVID-19. This study came out from Brigham, Boston, and earlier in 2020, September 3rd, looked at 18 consecutive patients with median age of 62, single-center autopsy study after COVID-19, and 11 of 18 were mechanically ventilated. And surprisingly and interestingly, all of the patients, 18 patients, had acute hypoxic injury in the cerebral cortices or cerebellum. And there were no other injuries found in this single-center study. And then note that no viral staining on immunochemistry study, IHC, analysis was found in 18 patients when they looked at 52 blocks. Let's look at another study from Germany. This is also autopsy case series. 43 consecutive patients, median age of 76, single-center autopsy study after COVID-19. 28% of patients were from ICU and 40% were hospital floorboards. They found that ischemic lesions were pretty common with 14%, and astrogliosis was 86%. And they said most commonly microglial activation and cytotoxic T cell lymphocytes infiltration were pretty common in cerebellum and brainstem. Note that it was actually brainstem and cerebellum in the infratentorial anatomical lesions. And SARS-CoV-2 viral proteins were found in 53% when they did immunochemistry analysis, and the locations were cranial nerves, lower brainstem. But they noted also these positive findings or proteins in the immunochemistry was not associated with any severity of neuropathological changes. Another study looked at detection of SARS-CoV-2 in autopsy. This one is 125 brain tissues samples from 25 autopsy cases. First, they looked at RT-PCR, which is the common way to test SARS-CoV-2, as you guys know. Not surprisingly, 91% were highly positive in nasal epithelium, and 72% had at least one very low but detectable positive CNS site by PCR. And the levels of detectable virus by PCR in COVID-19 brains were very, very low and did not correlate with the histopathological finding, just like the Germany study that I just presented. This study concluded that positive RT-PCR from this brain tissue were more likely from systemic inflammation more than direct invasion or infection, and synergistic contribution for hypoxia and ischemia, which is common in COVID-19 patients. They also looked at ISH assay, and immunochemistry, did not detect any viral RNA and protein in the brain tissues. You can see here in the schematic, they looked at CT, which is a cycle threshold. So in real-time PCR assay, a positive reaction is detected by accumulation of fluorescent signal. And the CT is defined as the number of cycles required for the fluorescent signal to cross the threshold. And usually less than 29 is a pretty positive finding. As you can see, nasopharyngeal swab and nasal epithelium CT or threshold less than 29, 16 to 29 is pretty strongly associated with staining here with nasopharyngeal swab and nasal epithelium. And as you go down here, more CNS tissue, frontal gyrus, temporal lobe, and cerebellum and medulla, you see less and less strength or intensity of cycle threshold, meaning that positive, pretty weak association or maybe contamination of the virus rather than direct invasion of or detection of direct infection of virus. And when you have positive findings in the CNS system, neuropathology findings here, or when you find the pathological findings in the brain, this is the breakdown. Hypoxia was really common. 41 out of 41, you see hypoxic changes, and the infarcts were pretty common, 44%. Hemorrhage was also 20%, pretty common. And you can see lymphocytic infiltrates and microglial activations were very, very common, which is non-specific, but these are the commonest finding you see with SARS-CoV-2. This is also interesting study published in Nature Neuroscience. This one also showed, compared to other studies, this actually showed ISH immunochemistry positive finding actually is similar to a prior one study. And they looked at PCR immunochemistry and ISH and electron microscopy. So different ways of analyzing the brain tissue. You can see this is olfactory mucosa OM positive blue triangle, and then CNS positive is the pink or red triangle. And you can see PCR in these different individuals. A lot of patients or individuals were positive for both CNS and olfactory mucosa when they try to detect the virus. And immunochemistry, less so to have both positivity, but you can see in different samples you have either OM positive or CNS positive. And ISH, you don't see any CNS infection. EM, no CNS infection, but only OM positivity. So among these studies that I presented, 4 or 5 studies, already you see a lot of heterogeneity among the studies, and some are positive PCR, but it sounds like a PCR is pretty sensitive marker for virus detection, but immunochemistry, pretty sensitive, but ISH, EM, not many studies showed positivity for SARS-CoV-2 when they looked at brain tissue. This is a systematic review, so there are a lot of papers published last year and this year with COVID-19 neuropathology. This looked at tissue-based neuropathological analysis papers. They found 101 cases or patients analyzed by PCR for SARS-CoV-2, and then 83 patients by immunochemistry analysis. And these were all autopsy studies except for 4 biopsy cases. On standard H&E slides, neither viral inclusions or specific cellular changes were noted as direct viral infection. That's pretty important to recognize. However, SARS-CoV-2 has been detected in the brain by its PCR immunochemistry and also electron microscopy when you look at different pathology studies. You can see 54% were PCR positive, 28% were immunochemistry analysis positive. Most common findings were microglial activation and lymphoid inflammation, different location, perivascular, parenchymal, or leptomeningeal. So these are pretty non-specific findings, though. And some studies reported 15%, quite a bit, microthrombi along with the viral detection. Again, I want to remind you different regions with SARS-CoV-2 detection. I showed you this figure, and if you summarize the current existing literature outside of CNS, SARS-CoV-2 virus was detected in cornea, conjunctiva, olfactory epithelium, bulb, and olfactory tubercle. CNS regions or border areas, frontal lobe, hippocampus, cerebellum, medulla, choroid plexus, cranial nerves, and trigeminal ganglion. So these are the common locations that were reported. I think lower brainstem, cranial nerves, and basal frontal lobes do make sense based on the discussion we had with transcribial entry and also bloodstream hematogenous spread for this virus. And however, we have to also remember there are a lot of limitations in these studies. They only reported or studied limited areas of brain because of COVID regulation, did not specify origin of cerebral cortex tissue when they reported cortex. And lack of control sample is a major limitation for these patients. Although you detect the virus or these pathological findings, there is really no control group to compare it to. And these are autopsy studies, so always there is a major hurdle when you look at autopsy studies. Hours later, when you look at the samples, there is also always a limitation. Different types of pathological findings I want to present. Inflammatory and reactive gliosis. We already talked about this in the schematic here. You can see perivascular lymphoid inflammation. As I mentioned before in the systematic review and other pathological studies, microgliosis and astrocytosis are two most common findings. And these are probably likely from systematic disease process rather than direct invasion of virus. We see this in sepsis, ICU illness. Any patients who are sick, you see this non-specific gliosis and astrocytosis with different locations of inflammation. And there is one study comparing control brains of sepsis, patients with sepsis, non-COVID-19, and COVID-19 patients, and they found a similar degree of microglia activation and perivascular lymphocytosis. So, probably these pathological findings are not that helpful, but this is commonly found in COVID-19 patients, just like other sick ICU patients. And microthrombi, infarcts, hemorrhages, hypoxic ischemia, these are important neurologic complications. As we know, strokes are common, and hypoxic ischemic injuries are common because COVID-19 patients have hypoxic ischemic brain injury, and also because of refractory hypoxia in these patients, especially with severe ARDS. So you can see in the graph, it's not too interesting, but eosinophilic Purkinje cells, you can see here in the top graph, and then this is a subacute infarct, and I believe this was a pituitary gland. So it just shows a subacute infarcts here. As we know, SARS-CoV-2 induces a cytokine storming, and it can cause severe hyperimmune reaction, and a whole activation of a coagulation system, and that leads to increases vascular permeability, and the endothelium gets really damaged. A lot of insult there, and we know that COVID-19 causes endothelial dysfunction along with the coagulopathy, and that causes also in the brain infarcts and hemorrhages. And as it turns out, SARS-CoV-2 can be detected within endothelial cells in the brain by immunochemistry and electron microscopy. You can see here microthrombi in the vessel, this pink tissue you can see, but when you look at this tissue in the frozen section, you can see this is a normal endothelium with COVID-19 infection. You see SARS-CoV-2 virus here and there. This is an infarcted tissue with ischemic stroke. You see more robust COVID-19 infection or SARS-CoV-2 viral detection in the patients with COVID-19. So when you have more stroke and ischemic tissue, it's not surprising that there's a BBB breakdown, and you detect more virus in the infarcted tissue. And this is showing that similar things you can see in the EM here, electron microscope, that you can see viral particles in various stage of bud formation, I have to say, and brain capillary endothelial cells showing various particles within cytoplasmic vacuoles. You can see in the arrow here, you can see viral particles in the endothelial cells. This particular case is in frontal lobe, and this is the only case I found that electron microscope showing that presence of viral particles in endothelial cells in the frontal lobe. And also non-specific finding, Alzheimer type 2 astrocytes. These are commonly found in hepatic encephalopathy patients, and these are commonly reported in COVID-19 patients. Although this is non-specific, I just wanted to bring you to the attention that these are commonly found in COVID-19 patients. In summary, so there is evidence that SARS-CoV-2 neuroinvasion can occur at the neuromucosal interface by transmucosal entry via regional nervous structures. And we talked about ACE2 and TMPRSS2 receptors. And we talked about medial structures of the brain, how they have abundant ACE2 receptors that may be the entry-level receptors for SARS-CoV-2 infection. And also we talked about the presence of SARS-CoV-2 in neuroglial cells of the olfactory tract and CNS regions border areas. And that's been identified with RT-PCR, ISH, and immunochemistry, and that different methodology really detected viruses in the brain. When detected, though, frontal lobe and lower brainstem appears to be the most common locations, and that goes along with probably theory that this can be transcribial entry and or bloodstream entry for these patients with COVID-19 CNS infection. Inflammation does not always coincide with virus localization, raising the concern that the virus may be evading the immune response in the CNS, which is probably the more common pathway. So based on the current evidence, direct infectious pathology may be possible, as well as indirect predominant, probably way of affecting the brain, immune-mediated neurodisruption. So probably systematic infection process, COVID-19 ARDS and ICU illness, or their critical care illness can cause immune-mediated response and causing neurologic complications. And although SARS-CoV-2 was detected in the brain tissue with different ways of detecting the virus, immunochemistry, PCR, or EM, cautious interpretation is needed because sensitivity of virus detection is suboptimal when samples are derived from autopsy samples. And also, COVID-19 patient, we know that the interval between the death and post-mortem exam is pretty long for these patients, and also samples are pretty limited. A lot of centers are getting this limited samples from the brain because of the COVID precaution. And also another point to make is a long COVID with persistent symptoms for a month, whether and how SARS-CoV-2 may persist in the brain needs further study and also research. And really the major limitation for these studies that I presented today is lack of control samples. And really additional studies, research with control sample, for instance, non-COVID-19 ARDS versus COVID-19 ARDS, looking at brain pathology with these different ways of detecting viruses, probably is necessary to better establish the frequency and the causality of direct infection in the CNS system. Thank you so much for your attention. Please let me know if there's any question. This is my email. Thank you again.